Okay, so how do I envisage the future of senescence research, particularly in vivo in tissue? I think the senescence field has focused on the tools that were available to us for decades, which were mostly cells in culture. Now, what we are realizing now is that there is a lot of heterogeneity, not only between different type of cells or different type of disease like cancer or age-associated disease, but also even within patient for example, in the context of cancer therapy, where we have a different type of or different cancer from different persons that will induce this cell aging phenotype in different manners. So this is actually raising a lot of very big challenges for in vivo senescence research. Some of them that I can identify is the ability to localize these senescent cells in time and place inside a tissue or in response to specific treatment. For example, when you irradiate a tumor, where are the senescent cells going to appear? What are they going to do? Are they close to the vasculature? Are they far from the blood vessels? So all these questions will need new tools that we are currently developing. One of the approach that we are using is spatial transcriptomics. So this is the ability of relocalize individual cells, and we are talking about very rare cells that could be just a few % of the total cells in a sample. So that's a very different concept from the research that we have done in the past using cell culture, where all of the cells in the culture undergo the same phenotype at the same time. Now we are able to track back these cells to a few % of the cell population in vivo. So that's one of the challenges, but most of these transcriptomic studies use a fixed moment in time, so a biopsy from a patient. Now the next challenge is to look at this over time, because we know that the tissue will change during the treatment or the therapy, and we don't exactly know when these senescent cells will appear. So another challenge in this context is to develop non-invasive biomarkers that we can watch over time. So it could be a molecule that is secreted from the senescent cell. It could be models with bioluminescence that allows us to track senescence in real time when it appears in the tissue. So yeah, I think we have a very exciting time ahead, and we're going to be able with these new tools to study the relevance of senescence in tissues and learn how to target them at the right moment in time and space.